Welcome and good day to all of you. Today in this video session, we are going to see about the topic membrane fluidity. The physical state of the lipid of a membrane is described by its fluidity or viscosity. In a simple artificial bilayer composed of phosphatidyl and phosphatidyl whose fatty acids are largely unsaturated. If the temperature of the bilayer is kept relatively warm at 37 degrees centigrade, the lipid exists in a relatively fluid state. At this temperature, the lipid bilayer is best described as a two-dimensional liquid crystal. As in a crystal, the molecules still retain a specified orientation. In this case, the long axes of the molecules tend toward a parallel arrangement, yet individual phospholipids can rotate around their axis or move laterally within the plane of the bilayer. If the temperature is slowly lowered, a point is reached where the bilayer distinctly changes. The lipid is converted from a liquid crystalline phase to a frozen crystalline gel in which the movement of the phospholipid fatty acid chains is greatly restricted. And the temperature at which the change occurs, it is called as transition temperature. Cholesterol, it acts as a bidirectional regulator of membrane fluidity. At high temperatures, it stabilizes the membrane and rises the melting point. At low temperatures, it intercalates between the phospholipids and prevents clustering. The transition temperature of a particular bilayer depends on the ability of the lipid molecule to be packed together, which depends in turn on the particular lipids of which it is constructed. Saturated fatty acids have the shape of a straight, flexible rod. Cis unsaturated fatty acids, on the other hand, have crooks in the chain at the sides of a double bond. Consequently, phospholipids with saturated chains pack together more tightly than those containing unsaturated chains. The greater the degree of unsaturation of the fatty acids of the bilayer, the lower the temperature before the bilayer gels. The introduction of one double bond in a molecule of stearic acid lowers the melting temperature almost 60 degrees centigrade. Another factor that influences bilayer fluidity is fatty acid chain length. The shorter the fatty acyl chains of a phospholipid, the lower its melting temperature. The physical state of the membrane is also affected by cholesterol. Because of their orientation within the bilayer, cholesterol molecules disrupt the close packing of fatty acyl chains and interfere with their mobility. The presence of cholesterol tends to abolish sharp transition temperatures and creates a condition of intermediate fluidity. In physiologic terms, cholesterol tends to increase the durability while decreasing the permeability of a membrane. Thus, cholesterol can be thought of as a buffering molecule in the membranes of cells that prevents abrupt changes in membrane fluidity over a range of temperatures. The importance of membrane fluidity. The membrane fluidity provides a perfect compromise between a rigid, ordered structure in which mobility would be absent and a completely fluid, non-viscous liquid in which the components of the membrane could not be oriented and structural organization and mechanical support would be lacking. In addition, fluidity allows for interaction to take place within the membrane. For example, the membrane fluidity makes it possible for clusters of membrane protein to assemble at particular sites within the membrane and form specialized structures such as intracellular junctions, light capturing photosynthetic complexes, and synapses. Because of membrane fluidity, molecules that interact can come together, carry out the necessary reaction, and move apart. Fluidity also plays a key role in membrane assembly. Membranes arise only from pre-existing membranes, and their growth is accomplished by the insertion of lipids and proteins into the fluid matrix of the membranous sheet. Many of the most basic cellular processes, including cell movement, cell growth, cell division, formation of intercellular junctions, secretion, and endocytosis, depend on the movement of membrane components and would probably not be possible if membranes were rigid, non-fluid structures. In this session, you have learnt about the topic membrane fluidity. Thank you for watching. Have a nice day.